Alrighty, thank you very much for that introduction, Dr. Lucchini. So as he said, my name is Danielle Coleman. I'm a student at the University of Illinois, and today in my first presentation, I'm going to be talking about the effects of rumen protected methionine supply to cows during heat stress and how this can alter liver tissue protein abundance of mechanistic target of rapamycin or mTOR signaling. And so just to start off with here quick, our overall takeaway message from this presentation is going to be that supplying rumen protected methionine during periods of heat stress may help to maintain hepatic homeostasis in mTOR, insulin, and antioxidant signaling. And so to start off with a little bit of background here, most of you probably know that methionine is often implicated as a limiting amino acid for our dairy cows. And in several studies with our transition cows, we've observed that supplementing rumen protected methionine can help to increase dry matter intake, milk, and milk protein synthesis. And those improvements, particularly in milk protein, have been linked to signaling changes in that mTOR pathway. However, this hasn't been fully studied in the liver tissue. Additionally, we know that methionine is going to serve as a functional nutrient, where we've observed increases in liver function and immune function in those transition cows. And so those effects are going to be best exemplified through 1-carbon metabolic pathways. And so this diagram here shows those 1-carbon pathways, and we're primarily going to focus here today on the methionine cycle and the transsulfuration pathway and the link to mTOR. And so just to walk you through quickly here, methionine is going to be at the top, and that can be synthesized here via homocysteine and can then be converted to S-adenosylmethionine. And then S-adenosylmethionine, or what we sometimes call it SAM, can be used to allosterically activate down here cystathionine beta synthase. This enzyme is the start of the transsulfuration pathway, and by increasing its activation, we can increase the production of 2 antioxidants, taurine and glutathione, and that can help to reduce oxidative stress and therefore inflammation. SAM is also used up here to activate a protein called SAMTOR. When SAMTOR is bound to SAM, we get activation of mTOR here, which can lead to greater protein synthesis. However, there's been very little work on the effects of supplementing rumen protected methionine during heat stress and how this can alter liver metabolism in our dairy cattle. We know that similar to the transition period, heat stress is going to be associated with a reduction in milk production as well as intake, leading to decreases in milk protein synthesis. And we also know during this time that our amino acids are going to be reprioritized away from milk protein synthesis. Moreover, this period of heat stress is going to be associated with increases in inflammation and oxidative stress, as well as a reduction in immune function. And so these are all going to be areas where rumen protected methionine supply during heat stress could be beneficial. And so our objective then with this portion of the study was to investigate the effects of rumen protected methionine supply during a heat stress challenge on the hepatic abundance of proteins involved in that mTOR signaling, as well as insulin signaling and antioxidant signaling. And so to do this, we used 32 cows in a crossover design at the University of Illinois dairy farm. And so we had 2 dietary treatments as well as 2 environmental treatments. And so for our dietary treatments, our first one was going to be our TMR, in which we supplemented that rumen protected methionine at 0.105% of the dry matter. And then our second is our control diet, which did not contain any methionine supplemented. And then our 2 environmental treatments. Our first one is going to be thermal neutral conditions, which will be called TN on our figures and tables. And then our second was heat stress conditions, which we induced using electric heat blankets, as you can see in this picture here on the left-hand side of the slide. And so to walk you through the study design real quick, it was divided into 2 periods, and then each period had 2 phases. And during that, when we changed to the periods, our environmental treatments were inverted. And so to explain that really quickly here, we see in period 1 that group 1 was under thermal neutral conditions for the entire 18 days. However, group 2 was under thermal neutral conditions for the first 9 days, followed by 9 days of heat stress. And then when we go to period 2, we see that this is opposite. Group 1 was under thermal neutral conditions for the first 9 days, followed by 9 days of heat stress, while group 2 was under thermal neutral conditions the whole time. And so we're going to consider this our sequence effect in our model. Another thing to note is that during phase 1, all cows were fed ad libitum. However, when we moved to phase 2, the thermal neutral cows were pair-fed to their heat stress counterparts, and this was to account for the decreases in dry matter intake that we knew heat stress would induce in our cows. For this portion of the study, we collected liver biopsies at the end of each phase 2, and then we took those to the laboratory and performed tissue explants, where we incubated tissue in media for 2 hours, and then used that to extract protein and perform Western blot analysis. For the statistical analysis, we performed all of our analyses in SAS using the mixed procedure. Our main effects were considered diet and environment, and then we also looked at the interaction of those two. And then to account for that crossover design, we included the sequence and period in the model. And so we'll move into the results here. Just to orient you to our graphs, they're all going to be set up the same way. We'll have our environmental conditions, thermal neutral and heat stress on the x-axis, and then our relative protein abundance on the y. And then our blue bars are going to be that controlled diet, while the orange bars are going to be the rumen-protected methionine diet. And so on this first graph, we have the environmental by diet interaction for phosphorylated mTOR, and this is going to be our active form of mTOR. And we see that rumen-protected methionine supply led to greater abundance of this p-mTOR under thermal neutral conditions, suggesting an increase in protein synthesis here. However, under heat stress conditions, we see a decrease in p-mTOR with rumen-protected methionine compared to control. So this suggests that potentially we have a shift in protein synthesis when we supply rumen-protected methionine during heat stress, and it's possible that methionine is being used for some of its other functions rather than protein synthesis here. On this graph, we have the abundance of phosphorylated eukaryotic initiation factor 2 alpha. This is also a protein involved in that mTOR signaling. And we see a tendency here for an environment by diet interaction. And so we see that the abundance here of PEIF2 alpha increased in our control cows during heat stress compared to thermal neutral conditions, suggesting that heat stress may be altering the initiation of our hepatic protein synthesis here. And then on this graph here, we have some of our other proteins involved in that mTOR signaling, and we have our main effects of environment and diet shown. And so what I want to focus on here is going to be the fact that we have an environmental effect here of heat stress on the ratio of phosphorylated 4EBP, which is our eukaryotic binding protein, to the total amount of that binding protein. And we see that this ratio increased under heat stress compared to thermal neutral conditions, again suggesting that we're changing some of our protein synthesis pathways when cows endure heat stress. On this slide, we have graphs for the abundance of total AKT on this first graph, and then the abundance of phosphorylated AKT. And so this is going to be a protein that is also involved in mTOR signaling, but insulin signaling as well. And so on this first graph, we see that the abundance of total AKT was increased by heat stress in our controlled diet cows. We see something similar for phosphorylated AKT, which is going to be our active version of AKT, where the heat stress cows actually on the control diet had greater abundance than all other treatments. And so this suggests that potentially there was a change in insulin signaling here, but we do want to explore this pathway further. This is going to be the start of kind of that insulin signaling. So we do have plans to measure other things such as insulin receptor and some of the transporters and that to fully understand whether this is actually a change in insulin signaling. And then lastly, on this graph, we have the environment by diet interaction for a protein called colin-3. This is going to be an inhibitor of a transcription factor, NFE2L2, which transcribes many genes related to antioxidant signaling. And so we see that the abundance of colin-3 was upregulated in our control cows during heat stress compared to our thermal neutral conditions. However, there was no difference due to rumen protected methionine under thermal neutral conditions, but it did tend to be lower than those control cows during heat stress. And so this suggests that potentially rumen protected methionine is helping to maintain our transcription of antioxidant genes and helping to maintain our normal oxidative status during heat stress. And so our overall preliminary conclusions from this work so far are that rumen protected methionine supplementation during heat stress may help cows to maintain hepatic homeostasis in mTOR, insulin, and antioxidant signaling. However, we do have more work that we want to do, and we do plan to do more measurement of proteins involved in mTOR, insulin, and antioxidant signaling to really dig deeper into those pathways. And then we also have plans to look at enzymes and metabolites involved in those one-carbon metabolic pathways, particularly at the enzyme level, to understand if we're really changing antioxidant production with the taurine and glutathione here. And so with that, I'd just like to wrap up and thank everybody who contributed to this project, particularly my lab members in the Nutriphysiogenomics lab, as well as members of the Dairy Focus lab here at the University of Illinois. And then particularly, I'd like to thank Adisseo for their funding of this project. And so with that, I'll take any questions that you guys might have.
Thank you, Danielle. That was an excellent presentation, and I hope the brains of the audience are not numb by now. It is a hard hit starting on Friday morning, but it was excellent, and I really appreciate your clarity in explaining all these effects. Just for clarification to the audience, this is a part of an overall work. There is a published paper already publishing the results of this, how the cows did and perform. And it is published on the Journal of Dairy Science. The reference is Journal of Dairy Science 2019, issue 103, page 2800, in case that you want to understand better the design and how the cows did. Daniel, I do have a question for you. You do have these signalings that apparently do have an impact on preventing milk fat or milk protein or the overall metabolic status of the cows. Do you, I mean, and you look into, obviously, I mean, methionine. Do you think that there might be other nutrients that may help on that, or more specifically, other amino acids that can help in this process?
Yeah, that's a great question and something that we've been kind of exploring. We think that there's potential for arginine to really have an effect here. It's also linked to that mTOR signaling as well as antioxidant status. And so we've been doing some work over the last couple years, myself included, looking at arginine and as well as a potential interaction between arginine and methionine. And so we actually have a couple abstracts that were presented this year at ADSA, really trying to understand the link there. And we're seeing that potentially there is some benefit so far at the mammary gland level is what my work is showing to supplementing both methionine and arginine.
Excellent. Yeah, I think this is a very hot and very interesting area of research. I do have a couple of other questions here coming in as we speak. One question is, do you think if methionine is oversupply can negatively affect insulin dynamics or insulin resistance?
Very interesting question. I'm not sure, to be honest. I would need to look into that a little more, but I don't think there's been a whole lot of work so far looking at if methionine is oversupplied in our dairy cows. And particularly at the liver level, I don't think we fully understand these potential effects here yet at the liver level. We don't really consider, I should say, hepatic insulin signaling as much as when we look more at like adipose tissue in that here in our dairy cows. So I do think that this does need more exploration, and the effects that we're seeing here are potentially really interesting because we don't necessarily think about looking at hepatic insulin signaling so much. So I think we would need to do some more work to fully understand if there would be any negative impacts there.
Okay. Well, homework to do and notes to be taken. And then I will just ask you another question, and then we'll move on with your second presentation, Danielle. But I think this is also a good question in terms of if there any effect of heat stress on the activation of mTOR, or heat state, or during heat stress can be led to the reduced phosphorylated mTOR.
Mm-hmm. So one of the hypotheses that we kind of have at the moment is that in this experiment, we saw an increase in phosphorylated or active mTOR with that heat stress. And we think that and the other changes that we saw potentially suggests at the liver level that we're increasing synthesis of some of our proteins. And I think this could be linked to our acute phase protein response here. We know that under heat stress, production of acute phase proteins is going to increase. And so this could be one of the mechanisms behind that change. And so one thing that we're hoping to do to try to understand this response with heat stress a little more is that I do have the media that we incubated these explants in. And so we hope to try and measure some of those proteins as well as some of our hormones and look at insulin in that supernatant fluid to try to understand further these effects that heat stress might be having on insulin signaling as well as the mTOR pathway.
Great, thank you, Daniela. For lack of time, I know that we have more questions, nice questions. I will, while you prepare for the next presentation, I will try to answer the couple of other ones that are more. So you go ahead and prepare for your next one, Danielle. There are a couple of questions relating to main impacts on the cows, temperature, and so on and so forth. And those results are published in this Journal of Dairy Science 103, page 2800. So Daniel, go ahead and go with your second presentation, please. Thank you very much.
Thank you. In this presentation, I'll be talking about the same experiment, but I'll be talking about the effects on whole blood mRNA abundance of genes related to the transsulfuration and antioxidant pathways. And so our takeaway message here is going to be that supplying rumen-protected methionine can enhance the antioxidant responses and transsulfuration pathway of whole blood. However, heat stress is going to have a negative effect on those pathways and responses. And so again, we are going to be talking about methionine, which many of you know is implicated as a limiting amino acid for milk production. And as I mentioned in the last presentation, when we supply rumen-protected methionine to our transition dairy cows, we have observed improvements in dry matter intake, milk production, as well as milk protein synthesis. And on top of those production responses, we do see effects more at a metabolic level. We do know that methionine is a functional nutrient, and it has been observed to promote liver function as well as immune function in our transition cows. And so those effects, again, are going to be best exemplified by our 1-carbon metabolic pathways. And so we see those pathways here on this diagram again. Up here this time we have the methionine cycle with the transsulfuration pathway being down here at the bottom. And so we'll go through this quickly again. We have methionine here, which can be synthesized from homocysteine via 2 enzymes, MTR and BHMT. And then in these pathways, methionine can then be metabolized to produce S-adenosylmethionine, or SAM, which again is our universal methyl donor. Now SAM again has a variety of functions, but what we're looking at here is its ability to allosterically activate cystathionine beta synthase and thereby increase flux of this transsulfuration pathway down here. And so by increasing flux of that pathway, we can increase production of both taurine and glutathione, which are 2 of our major antioxidants. And so in this way, methionine can help to reduce oxidative stress and inflammation in our dairy cows. However, while we know rumen-protected methionine can have these effects during the transition period, there's been very little work with it related to heat stress, and particularly in this case looking at the inflammation and oxidative stress that are associated with heat stress. And as I mentioned in the first presentation, we know that during heat stress, feed intake is going to be reduced, leading to a decrease in milk production, similar to what we see in the transition period. And so because of this, methionine could have a potential role in promoting production during heat stress, as well as helping to reduce some of that inflammation and oxidative stress, as well as improve immune function in our dairy cows. And so, therefore, our objective with this portion of the experiment was to investigate the effects of rumen-protected methionine supply during a heat stress challenge on whole blood mRNA abundance of genes related to immune and antioxidant responses, as well as that transsulfuration pathway. And so this will be the same design as before. We used 32 cows in a crossover design. Our 2 dietary treatments, our first one was our TMR with that rumen-protected methionine at 0.105% of the dry matter. And then our second was our control diet, which was that TMR without rumen-protected methionine. And then we also had our 2 environmental treatments. Our first one will be thermal neutral conditions, which will be called TN on the tables and graphs. And then we also had our heat stress conditions, which we induced using electric heat blankets, which you can see again here in this figure. And then because this was a crossover design, again, we had 2 periods and 2 phases per period. And so cows remained on the same diet for the entire experiment, but what changed between the periods was their environmental conditions. And so just to explain that again, using group 1 and 2 as an example, we see that under thermal neutral conditions, group 1 was exposed to those during period 1, but during period 2, they were exposed to heat stress for the second 9 days. And we see group 2 is opposite of that, where they experienced heat stress in period 1, but did not experience it in period 2. And so we're going to consider this our sequence of environmental treatments, and this will be included in our model. Additionally, a thing to note is that during phase 1, all cows were fed ad libitum. However, during phase 2, our thermal neutral cows were going to be pair fed to those heat stress counterparts in order to account for decreases in dry matter intake that we knew heat stress would be causing. And so for this portion of the experiment, we collected blood approximately 4 hours post-feeding on day 8 of each phase 2. We then took that blood to the lab and extracted RNA, and then we performed PCR. For the statistical analysis, we again used a mixed model in SAS for all of the analysis, and our main effects were considered our diet and environmental effects, as well as their interaction. And then we also included sequence and period again to account for that crossover design. So now we'll move into the results. Again, we'll have our environmental treatments on the x-axis and our relative mRNA abundance this time on the y. And then our blue bars again are going to be our control diet, and the orange bars will be the rumen-protected methionine diet. And so on these first two graphs, we have our interaction for two genes in the transsulfuration pathway, CBS again, and then cysteine sulfonic acid decarboxylase, which is also involved in the production of taurine in that pathway. And so in both cases here, we see that our rumen-protected methionine cows had a greater abundance during thermal neutral conditions compared to heat stress conditions, suggesting that there is some changes with rumen-protected methionine in the synthesis of taurine. On this table here, we have the main effects of environment and diet for some of the other genes in that transsulfuration pathway. And the ones I want to point out here are going to be the mRNA abundance of cysteine dioxygenase 1, cystathionine gamma lyase, here as well as glutathione synthase. And so for the first two, CDO and CTH, we see a downregulation in their abundance with heat stress compared to those thermal neutral conditions. And then we see a tendency for the same effect here with that GSS. And so this really points to the fact that heat stress is potentially going to be downregulating our transsulfuration pathway and leading to less production of our antioxidants. Looking at genes involved in our antioxidant response, on this graph we have superoxide dismutase 1. And we see here that our rumen-protected methionine cows had a greater abundance of this enzyme during thermal neutral conditions compared to heat stress. However, there was no difference under heat stress here between the control and rumen-protected methionine diets. And then on this last graph here, we have the interaction for a gene involved in immune responses, toll-like receptor 2. And we see a tendency here for that environment by diet interaction. And we see that the mRNA abundance of this TLR2 was increased in control cows during heat stress compared to thermal neutral conditions, really hinting at the fact that heat stress might be upregulating our immune responses during in the whole blood, and so this could really be contributing to increases potentially in inflammation, as well as some dysregulation of the immune cell function. And so overall, so far, these results highlight the negative effects of heat stress on the whole blood transsulfuration pathway and antioxidant responses. They also highlight the fact that rumen-protected methionine may enhance the antioxidant responses in the transsulfuration pathway of whole blood, helping to potentially reduce oxidative stress. However, we don't see those effects of rumen-protected methionine during heat stress. And so we do plan to do some more work with these samples as well. We plan to measure some of the cytokines in plasma from these cows at this same time point. And then we also want to look at the mRNA abundance of other genes involved in 1-carbon metabolism to really dig deeper and understand metabolism by methionine during heat stress. And so with that, again, I'd like to wrap up just by saying thank you to everybody that contributed to this project, especially the members of my lab group in the Nutraphysiogenomics lab, as well as the Dairy Focus lab at the University of Illinois. And then again, a big thank you to Adisseo for their funding of this project. And with that, I'll take any questions related to this presentation as well.
Thank you, Danielle, for another excellent presentation and giving us all a big headache on a Friday morning. I just for the lack of time, I do have one question for you, and then we'll move on with the next presentations. But just to put all this work down to earth, what would you suggest? Cows, whenever they hit stress, obviously we know what happens, and then it takes a long time for them to recover, particularly in meat production, meat composition. What would you suggest to a producer to use any nutritional strategy to help ameliorate this effect or help to cure it after it happens?
I think it would be very beneficial. I think anything we can do to really improve those cows and help them recover faster, it can really help the cow to be healthier and have better welfare, but it can also help the producers economically. And I think that methionine in particular could be really important there. And I think that's another step to potentially look at in the future, is to really understand, is this having effects after heat stress, helping those cows recover.
Excellent. Well, thank you, Daniel, for taking the time and doing these presentations. For lack of time, or in respecting the time for the other presenters, we're already 5 minutes behind. I will introduce the next presenter, and the next presenter is Jorge Carneiro. He is down south, very much down south in Brazil. He is a student of Dr. Rodrigo Almeida. And I will just let Jorge to move on with his presentation. Thank you.
Thank you, Daniel. Thank you, Brian, and all the Adisseo group for the opportunity. And my presentation is working?
Yeah, now it is, Jorge.
Yes, okay. Well, my name is Jorge Henrique Carneiro, and I am a PhD student in the Universidade Federal do Paraná in south of Brazil. And my advisor is Dr. Rodrigo Almeida. And we are going to talk a little bit about our project that has the name Effect of Rumen Protected Methionine Supplementation and its relationship with lipids metabolism in high producing dairy cows with different dietary energy concentration. When you look at the previous studies with a high methionine concentration in the diet, in addition to the constant effect in milk protein content with methionine, we realized that in some studies there were a milk fat increase also, and in particular in postpartum trials. Besides that, we observed in some studies that the cows became a little bit more efficient to carry over the consumed energy to the milk, for example. And recently, some studies has shown a post-absorptive effect of methionine in milk fat synthesis in mammary epithelial cells. Several pathways has been suggested to explain this response, but the main point seems to be linked with SRBP1, for example, and its respective controls like CRCT2 and another protein controls. In this context, our project has the main point to evaluate the possible effect of a high methionine intake in the lipid metabolism of high producing dairy cows at the peak and mid of lactation in two different concentrations of energy in the diet. As a second point, we would study a possible effect on the ability to export lipids to whole body with a high methionine intake, and also a possible effects on de novo synthesis of fatty acids in the mammary gland. To do this trial, we used 112 hosting cows with 49.4 kilograms of milk and 146 days in milk on average. We blocked the cows by lactation number, milk yield, and days in milk. The experimental periods lasted 38 days, and we used the first 3 days as a covariate. The TMR was offered once a day, with or without the high energy treatment on the wagon, on the TMR. And the methionine treatments was offered twice a day on top dress. Milk samples was collected weekly, and in the last 9 consecutive milkings, we used it for composition, milk fatty acid profile, and to filter gene expression. All the data was analyzed and mixed, proceed of the SAS with repeated measures. The experimental diets was based in a typical diet of South Brazil, based in corn and soy. And so the first treatment was a control diet. The second treatment was a supply of methionine, then you use smartamine then. And our point was to match 2.4% of methionine and metabolizable protein. The third treatment was a high energy treatment, and we used hydrogenated fat to do this. And the fourth treatment was a combine of high energy and methionine. We used these products, a hydrogenated fat, to increase the energy in the high energy treatment, because we don't want to change the fermentable carbohydrate in the rumen, and also we would to get to the cows a fat that has a similar profile of fat than the body fat, and it has the smaller risk to change the biohydrogenate of fat in the rumen. In this diagram, we can see when each procedure was done during the trial. And here is the preview results. In the preview results, we can see that high energy get more milk, but without any increase in content of the milk in fat or protein. But methionine was able to increase milk fat content and milk protein content with an interaction between energy concentration of the diet and protein content and protein yield. But we observe a reducing in lactose content when we increase the supply of methionine. And when we get more energy for these cows, the lactose increase again. The energy correct milk was not different between high energy methionine and high energy plus methionine. And we observe a little increase in milk urea nitrogen with high energy treatments. When we look to the behavior during the time, the milk protein content and the milk fat content has a constant increase during the period, and the treatment with methionine was more constant in energy correct milk, for example. When we look the milk energy output, we can observe that the high energy methionine and high energy plus methionine treatment had no difference, was the same. And this can just suggest for us that maybe the cows just change the place when they put the energy in the end of the day. As a conclusion, the greater supply of methionine show potential to change lipid and protein metabolism, interaction between energy concentration and methionine on total milk protein, as well additive effect on milk protein production. And we need more analysis to the fatty acid profile of milk and the possible pathways of de novo synthesis in the mammary gland to be better understand the mechanisms involved between methionine and lipids metabolism. Well, thank you for the time, for the opportunity, and I hope then I could answer some questions.
Thank you, Georgie, for your good presentation. Excellent results. Very interesting data. Just have one question. Did the cows fed just the rumen protected methionine by themselves? Did they lose body weight?
Yeah, no, I don't show this data, but no, the body weight was not affected with the treatments.
Okay, thank you. Well, the basal diet had a good dose of fat. It was a 4.3 the control diet compared to the diet with the hydrogenated fat. Do you think that if the fat content of the basal diet would have been less, would you have seen these effects?
Well, I think that the point is this fat was helping the cows to increase again the milk yield, for example. When we observe the treatment with methionine, the milk yield was a little bit lower than the other treatments, and I guess that the cows expend more energy, for example, to synthesize fat and protein, and this the cows then has methionine and energy get work with this better, I guess. I mean, I don't know if we got less fat if the cows had the same results. I guess maybe not.
Okay, thank you. And then I see that did you you are showing here, and I'm going to kind of set up Andrew's talk. You did set up here the grams of methionine per the methionine per metabolizable energy. Oh, you have there for the lysine as well. Okay, yeah. I got it. Okay, yeah, okay. Okay, well, thank you for your excellent presentation, Georgie, and thank you, Rodrigo, also for your work. I'm looking forward for the following up results on this trial. The next presenter it's going to be Andrew Lapierre. He is a PhD candidate at Cornell University under the supervision of Dr. Mike Van Amber. So I will let Andrew go on with his presentation. I think Georgie's presentation was a nice setup for Andrew's. Good morning, Andrew.
Yes, good morning. Good morning. Thank you, Daniel, and thank you, Jorge, for the great presentation. So today I'd like to talk a little bit about some of the work that we did in the last few months with the Adisseo group, discussing digestible amino acids in dairy cattle, their efficiency of use, and what happens to that efficiency of use when we vary dietary carbohydrates in the form of starch. So to start, my work is interested in the interrelation between the supply of amino acids, particularly essential amino acids, and the metabolizable energy in which those dairy cattle receive within their diet. And so that really stands on the shoulders of a previous PhD student who did his work with the updated version of the CNCPS. And evaluating what happens when we do relate supply of amino acids relative to metabolizable energy. And so through some data set corroborations and generating some results, he ended up finding some what we deem optimum supplies of essential amino acids relative to metabolizable energy. And this is a common practice that we see with some of our folks over in the swine industry relating standard ileal digestible amino acids relative to the energy that those sows are being fed. And so this isn't necessarily novel, but we are looking to relate this relative to energy. And so with that in mind, that whole principle allows us to look at some amino acid balancing relative to that grams of essential amino acid relative to energy, which would permit us to feed lower MP diets while maintaining lactation performance. And we've done this in several studies already through help with Adisseo. So it is possible we are able to shave down some nitrogen that's being fed and more precisely implement maybe some RP products that allow us to dial in our methionine and lysine contents within those diets. But to further expand upon that, there has been some literature that would suggest that if we were to alter the supply of any gluconeogenic nutrients, whether that be propionate through VFA production or actual intestinal glucose presence, we do see alterations in milk protein output, relatively speaking, through upregulation of IGF-1 and the somatotropic axis pathway. So there might be some opportunity there if we were to influence the supply of gluconeogenic precursors to influence our efficiency of use for those essential amino acids. Some studies have been done by increasing gluconeogenic supply, but that's typically been done through abomazole infusion. And when it's not done through abomazole infusion, the range in which the inclusion for dietary starch or non-structural carbohydrates is typically out of the range in which we would see in our North American diets. So with that in mind, our goal was to actually vary non-structural carbohydrate levels within a typical range that we would see in North American diets, and then evaluate what happens when we test those levels of amino acids relative to metabolizable energy. So we conducted a study from December to April of this past year. Was conducted as a pen study to mimic more of what we would see out in the industry with 4 different diets. It's a 2x2 factorial design. And we fed it to 3 pens per treatment. And within those pens, there were 16 cows, so a total of 192 cows with a 25-75 split of primiparous and multiparous cows. And we did want to make sure we were balanced within pens, so cows were blocked by parity, body weights, and previous milk production, as you can see that there. The treatment outlines are as follow. We had 2 levels of dietary starch inclusion, as well as 2 levels of supply for essential amino acids relative to metabolizable energy. So you can see on the left-hand side, we have our LS100 and our LS105, both of which are being fed or formulated for 23% starch on a dry matter basis. And then the 100 would designate the 100% grams optimum, which is what you saw in the first slide, trying to target those ratios there. And then the 105 would be a 5% increase of those ratios to see what would happen if we were to titrate in a little more supply, if we would see some greater protein output in those cattle. On the right-hand side, we have a similar designation, just that we have an increased formulation of dietary starch at 29% dry matter with the HS100 and the HS105. We did want to make sure we're testing amino acids here, so we did want to be isocaloric for all 4 diets, varying only in dietary carbohydrates and amino acid supply, and we did formulate these diets using version 7 of the CNCPS. We had a 10-day covariate for all pens. They were all fed the HS100 diet to get baseline levels for milk production as well as blood metabolites and total tract digestibility. And then the pens were assigned to one of those 4 treatments for 50 days. We took daily dry matter intakes and milk yields, weekly body measurements, milk composition, plasma samples every other week for PUN and plasma levels of amino acids, as well as feed ingredient, TMR, and refusal samples on a weekly basis. Again, we used 192 cows, 12 pens, 4 dietary treatments, and given some of the constraints that we had at the farm, we had to split this up into 2 longitudinal enrollment periods for these cattle. Oh, and then we also had 3 fecal spot samples for total tract digestibilities for those 4 diets. All stats were analyzed using SAS statistical software. We had descriptive feed and TMR samples and animal performance, under the notion that our block or pen within enrollment was used, and then the main effects of starch level, amino acid level, and time, and we use interactions of those 3 when appropriate. And then we included the random effect of cow within pen. And then whenever available, we use covariates. And because this was a longitudinal study, we used repeated measurements with Tukey adjustments for those treatment differences. A breakdown of the 4 diets you can see here on a dry matter basis. And what I really would like to point out is that in order to have differences in starch levels and non-structural carbohydrates, we did include steam flake corn and cornmeal at higher inclusion rates for that HS100 and HS105 diets. But to maintain isocaloric nature for all 4 treatments, we did have to include a high POM energy booster into the mix for the LS and LS105. We did utilize some urea to maintain rumen N balance, as well as the inclusion of some smartamine M and ML to really dial in the methionine and lysine levels for these diets. From an observed chemical composition for these 4 diets, you'll notice that crude protein was different as expected for the 100 and the 105 groups, ticking up for the 105 groups relative to the 100 groups. Unfortunately, looking at the starch levels, we did see a relatively close 23% dietary starch for the LS 100 and 105 group, but we didn't quite get to the 29% for the HS 100 and 105. But with that in mind, we kept moving forward. Our MP supply here for essential amino acids looks pretty similar to what we would expect when we have the 100% supply relative to the 105. So particularly looking at the lysine and methionine here, we did see the trends going up as we move from 100 to 105. Our ME, as you can see on the bottom, was nearly identical for all 4 treatments. And then when we relate methionine and lysine relative to the metabolizable energy, you can observe those numbers there. Looking at some of the results for our dry matter intake, we did see some differences, relatively speaking, between the high starch and the low starch diets, with the low starch diets having lower levels of dry matter intake. We do believe that's because of some differences in forage inclusion, as well as the different types of fiber that we were feeding these cattle. So the HS 100 and 105 groups did have increases in soy hulls and other more highly digestible levels of fiber, which would have allowed for greater or quicker passage and digestion so that their rumen fill wasn't always met. Looking at energy corrected milk, the HS 105 group was the highest out of the 3. But what we did notice was that the energy corrected milk for the amino acid level at 105 was higher relative to the 100 group. We do believe that's because of different reasons, one of them being that the true protein yield was greater when we look at the high starch diets, whereas when we look at the fat yield, it's greater for the low starch diets. Now, we do think the greater protein yield is because of the starch effect. However, the low starch diets having the greater fat yield may be attributed to the greater supplementation of preformed fatty acids in the form of that high POM of fat. So lactose yield was a little bit different, but that would be indicative of the changes in milk yield. And then the milk composition for protein, fat, and lactose does follow suit for those yields. Body weight and condition score didn't change for any of the 4 treatments relative to the initial or the final body weight as well. We look at efficiency results, so milk yield and energy corrected milk relative to dry matter. We do actually see greater efficiency in the low starch groups relative to energy corrected milk. Again, we do think that is partially an artifact of the hot energy booster that we did feed to these cattle. Whereas when we do see the HS100 and 105, they're not far off, but they are statistically different than the low starch group. From a nitrogen efficiency standpoint, however, we do see that the lower levels of amino acids were actually more efficient with their nitrogen. So the LS100 and more importantly, the HS100 had greater nitrogen efficiencies, both around 33%. MUN levels, again, you can see there lowest on the LS100 and highest on the 105 level. We do think that's because the LS105 didn't quite have the fermentable energy it needed to utilize the extra amino acids that we fed those cattle. So in conclusion, increased levels of dietary starch had greater milk protein percentage and yield. However, the lower starch diets had greater milk fat percentage and yield. So that would be attributed to why we see those differences in energy corrected milk production for the LS105 and the HS105 group. Again, cattle fed a lower level of dietary starch consumed less dry matter, but that was likely due to the greater inclusion of forage in the diet. And it did appear that cattle on the lower starch diet reached physical fill based on fiber levels, but we are looking at that further. Feed efficiency was improved for cattle on low starch diets, but again, we partially attribute that to inclusion of high palm fat. But I think most importantly, and the really goes with the hypothesis of the study, is that the optimum amino acid per mcal of metabolizable energy, so that 100% had the greater nitrogen efficiency, with the high starch 100 diet having the greatest nitrogen efficiency of all. So there is more work that needs to be done. Juggling everything with COVID and stuff here on campus, we need further feed chemistry and laboratory analysis to help clarify these results further. So with that, I'd like to acknowledge my PI, Mike Van Amberg, and everybody around us in the Cornell team, the Cornell Ruminant Center team for their help with the experiment, and of course, Adisseo for the financial and intellectual expertise. So with that, I'll take any questions.
Thank you, Andrew, for your excellent presentation. I have I have one question. What time did you take the blood samples?
Yes, good question. Blood samples were taken for every time point. They were taken 8 to 10 hours post initial feeding. So feed was usually dropped between 6 and 7 in the morning, and then when they returned from milking, about an hour after that, we took samples from them in the afternoon. So it would be about 2 to 3 p.m. in the afternoon.
Okay. Do you think does it matter if you're in your gluconeogenic diet? Does it matter if it was glucose or propionate?
Yeah, I think it depends on which form that you are feeding. Particularly in this study, we were really going after a propionate response to try and drive not only an insulin or IGF-1 response, but also microbial yield to really help with that protein output. I do think they have similar pathways in which they work, being propionate versus glucose. But I think for this study, we were really interested in dialing in that propionate response.
One more. Well, I have two more questions. In the high starch diet, obviously was lower fat. Do you think that because those cows produce less milk fat, do you think that that was could have been because of a lack of long chain fatty acids precursors to express the milk fat of those cows?
Yeah, so this, the supplementation of a high palm fat was to help with, obviously, with the isocaloric natures, but as it has become increasingly clear, the supplementation of the type of fatty acids that you're giving these cattle will have an effect on how they partition particular nutrients. So we have been working with a collaborator here on campus, Dave Barbano, on taking some of the milk that we sampled from these cattle and really looking at the fatty acid profile from these cows and really seeing if there is a response in the amount of preformed or de novo or mixed fatty acids, and then how that may or may not have affected the efficiency of use of these amino acids that we gave these cattle.
Wonderful. And last question, just for lack of time, but one down to earth. Which one of these 4 diets would you feed to those cows?
So I think, given everything we've got here, we really, given circumstantially the way milk protein is these days, I would really try to dial in on that HS100 diet, because that really, we're trying to feed for that optimum. We're really trying to hone in on microbial yield and really try to get a propionate response for these cattle. So that HS100 diet is likely where I would go. There may be some opportunity to maybe push it towards that 105, but again, I think that's dependent upon feed availability and financial resources for those diets.
And by 100, you mean not only lysine and methionine, but all the amino acids, correct?
Yeah, so thank you. That's an important distinction. Yes, we focus on lysine and methionine because they are most limiting. But again, with these studies, this one that I just presented, as well as some others that we've done in the past, we're really trying to dial in all essential amino acids. So not just titrating in different levels of lysine and methionine, although having products, rumen protected products, does certainly help get us to where we really need to be for those levels of methionine and lysine.
Yeah, I asked that question because I know how hard you have to work to make those diets. So.
Yes, there's a lot of moving parts there. Yes.
Yeah. Well, thank you, Andrew, for your presentation. And for lack of time, I need to introduce you guys to the next and last presenter for today's webinars. For today's roundtable. The next presenter is Dr. Kerry Harbatin. I don't think he needs much more introduction, but he is obviously a professor of nutritional physiology department in the Department of Animal Sciences at Penn State University. With that, I will let you going into Kevin's presentation. Thank you.
Thank you for having me here to talk about our experiment where we're extending past some of our previous work that we had done with HMTBA. So biohydrogenation-induced milk fat depression, we don't need to talk about that very much, but it's a really common cause of reduced milk fat. It doesn't cause every change in milk fat that we see on a herd, but it is a common cause. Methionine is supplemented in different ways. So we have HMTBA, that's a rumen available form, HMBI, that's partially rumen available, and then we have rumen protected encapsulated synthetic methionine. So we've previously shown in a number of experiments that HMTBA supplementation reduces the shift to the alternate biohydrogenation pathway and reduces that induction of milk fat depression when we're feeding low fiber and high starch and unsaturated fat diets. Just quickly wanted to show a little bit of that data. So this is our first experiment with HMTBA, where we had low and high producing cows, and then we fed diets that were low, medium, or high risk for milk fat depression. So basically what's interesting with milk fat depression is that we know what our risk factors are. We know high producing cows are at higher risks than low producing cows. We know that as we increase starch and increase unsaturated fat or a number of other risk factors, we increase the chance of dropping into milk fat depression. So we're able to experimentally create situations where we have higher risk. So in this experiment, the blue is control, red line is HMTBA. You can see the low producing cows didn't have much trouble, that they maintained pretty normal milk fat as we went from that low, medium to high risk diet. Our high producing cows, on the other hand, as we went to a medium and high risk diet, the control cows dropped rapidly in milk fat. So this is a 0.7 unit difference in milk fat during the high risk phase, while our HMTBA cows were able to maintain higher milk fat. When we look at our trans 10 isomer, we can see that our control cows in blue in the high group had a really big increase in trans 10 as we went to that medium and high risk diet. So this really shows that there's a ruminal effect of HMTBA. We have a recent paper out, collaboration with Dr. Pita at UPenn, where we show that there's a microbial population change. So we're maintaining increased diversity in the microbial population when we have HMTBA, and there's a lot of thinking that diversity is really important to the microbial population and maintaining normal fermentation. There must be some of those populations that we're helping sustain in face of this high starch and high unsaturated fat. They're important for biohydrogenation. So our objective in the current experiment was to compare HMTBA to other sources of supplemental methionine when feeding diets with increased risk for milk fat depression. We had 10 primiparous and 26 multiparous cows. This is done in Kaylin Gates, and we had a 5x5 Latin square. So the experiments get really difficult for me to follow once I get beyond 3 treatments. So I have to really concentrate remembering what all of the treatments are. I always tease Lou Armitano. He designs these 5x2 experiments, and it makes my head explode. We have a little bit of that, but we really wanted to get at these differences in our different sources of methionine. So we have control, which is just our corn carrier. We have HMTBA, which is targeting 30 grams of HMTBA, HMBI, 43 grams of HMBI from Metasmart, HMTBA plus HMBI, so we have 12 grams of HMTBA and 26 grams of HMBI, and then we have rumen protected methionine, which is 26.5 grams of smartamine M. And then to make this even more complicated, we have different dietary phases, because basically what comes down to that if you expect to your treatment's having a difference during milk fat depression, you have to induce milk fat depression to see that difference, right? So if we go back to that previous data I showed, that if we just did that experiment on safe diets, like a lot of our experiments are done, we don't give the opportunity for a product to actually show what it can do in a risky situation or when that cow's starting to get challenged. So day 1 to 20, we have low risk for milk fat depression. This really allows diet adaptation also. Day 21 to 27, we go to moderate risk for milk fat depression. So we're dropping forage NDF. In increasing soybean oil in the diet. So this is mimicking rapidly available unsaturated fat. Day 28 to 30, we go to a high risk of milk fat depression, so we decrease forage NDF even more and increase soybean oil. And then we make observations across these phases. Of course, the cows don't always read your experimental plan and hypothesis, and sometimes they don't react exactly as you would like. We started this experiment, I believe it was in late November or early December. We had to start with our new crop corn silage, so it was not as long through the fermentation process. It happened to be drier corn silage. and we had a harder time inducing milk fat depression. So in period 1 and period 2, when we went into the moderate and high risk situations, we didn't see very much of a decrease in milk fat. So we made a little bit of an adjustment to these moderate and high risk diets. We made them a bit more challenging, and we did get more of a decrease in period 3 and period 4. But in period 5, we actually had those cows are getting further out in days in milk, and then we didn't have as large of a response during the induction in the moderate and high risk phases in period 5. So a little bit, when you look at the data, there's a little bit of a difference in the overall magnitude of those inductions during those periods. We did test for that interaction, and I'll come back to that in a second. So our model, we have a random effective cow period, fixed effect of treatment, milk fat depression risk during the period, our parity in their interactions, repeated observations of day, and subject of cow by period. So now you can understand why it gets really complicated to try to keep all this data in your mind at once. Luckily, we had no 2 or 3-way interactions of treatment and parity. So I'm not going to show any of the parity data today because there weren't any interactions there. We also tested for this block effect. So block of period 1 and period 2, and then period 3 to 5, where we changed the magnitude of those inductions, and we did not have treatment by block interaction. So that allows us to look at all the data together. So I would say is that even though there's a slight difference in the magnitude of that decrease in milk fat that we're getting between these periods, overall it did not change what the treatments did, right? So we're able to look at those main effects, but we were modeling out those impacts. Milk yield was increased by supplementation only on day 13. So just to kind of orient you to my format and my data, in the upper right-hand side, we have the overall effective treatment day and that treatment by day interaction. So we had treatment by day interaction for milk yield. And then the first line in the data is the overall effect across all of our experimental days. And then we look at the data by day. And then our p-values, we compare HMTBA column is control versus HMTBA, and same thing for all of the others. We're comparing each of the treatments back to the control to say, are they having an impact? So if we looked at that overall effect, there's a tendency for HMTBA to increase milk yield, but this impact really was coming from day 13. So on day 13, we have a significant increase in milk yield for HMBI and the rumen protected methionine. We have a tendency for HMTBA and HMTBA plus HMBI. Then there's a tendency for, or actually I think this is significant at just under p.05 for HMTBA on day 20. This has confused me a little bit on why we saw this on day 13 and not on the other days. You know, at first I was kind of thinking maybe this is just something that popped up as being on that day. But remember, this is a 5x5 Latin square, so this day 13 is replicated over 5 different periods. So this isn't that it happened to be a change in the silage or something like that, a day effect that way. It is replicated. And I'll show later, we also have some changes in milk protein on day 13. So I think what we might be seeing here is some sort of a transient adaptation that's occurring to these diets during that phase. I think that's interesting response. Milk fat was our primary interest, so we have an overall effective treatment, effective day, no treatment by day interaction. It is down to 0.16, but it's not significant. So if we look at our overall effect, controls 3.62 across our entire experiment. HMTBA is increasing. HMTBA plus HMBI is significantly higher, and rumen-protected methionine is also significantly higher. The HMBI is not; it's numerically higher, but it's not reaching significance. If you look at our decrease, day 20, these cows are at 377. We're taking them down to a 3.54. So we're getting a small decrease in milk fat percent. And our responses are mostly at that medium and high risk phase, and that's what we would expect that our product's able to have an effect when we start into those challenging situations. Milk fat yield is a similar story, that we have an increase with HMTBA, HMTBA plus HMBI, and rumen-protected methionine. Here on the yield, that increase in milk yield on day 13 is driving a significant increase in milk fat yield on day 13. And then we also have an increase in milk fat yield on that medium and high risk diets on day 27 and 30 that's being driven by that change in milk fat percent. So looking at trans 10, 18.1, this is our best indication of that alternate biohydrogenation pathway. We have no effective treatment, effective day, and no treatment by day interaction that's significant. But when we look at the data, with our pre-planned contrast, we do have a tendency for HMTBA to be lower than control, and the response is mostly on this day 27. So we are increasing trans 10 from 0.45, which I consider normal. 0.4 to 0.6 is about where we normally see herds. So we're increasing that 2 to 3 fold, so getting up to 1.24%. So we're getting a modest amount of milk fat, trans 10 type milk fat depression. Our HMTBA is significantly decreasing on that day 27, but not as large of an effect in trans 10 intermediate as we had seen in our previous experiments. De novo fatty acids, so normally we see de novos decrease during diet-induced milk fat depression. We do have a day effect, so we are decreasing these de novos, but we're not having any effect from any of our methionine supplements. And this is a little bit unexpected, because in our previous data, we saw an increase in de novo. So it seems like the changes in milk fat that we're having in this experiment are not simply from alleviating a classical diet-induced or biohydrogenation-induced milk fat depression. Looking at milk protein data, so there's an effective treatment and a treatment by day interaction. Rumen-protected methionine is increasing milk protein percent. There's a tendency for HMTBA plus HMBI. And as I mentioned before, this effect is most significant on day 13. So all of our methionine supplements increase milk protein, not a lot, but they increase milk protein on day 13. Rumen-protected methionine was increased or tended to be increased on all days. Milk protein yield, we have a tendency for an increase in milk protein yield for rumen-protected methionine. Again, that day 13, everybody's increasing milk protein yield, being driven by that increase in milk yield. And rumen-protected methionine is more consistent in that increase. So summary and conclusion, our challenge resulted in only a modest decrease in milk fat, so we're decreasing to about a 3.5%. But trans 10, 18.1 was nearly tripled. HMTBA, a mixture of HMTBA and HMBI and rumen-protected methionine resulted in increased milk fat concentration and yield. The mechanism appears to be independent of trans 10 under these circumstances, and may be due to acetate supply or post-absorptive mechanisms. So, trans 10 isomers are not the only thing that impact milk fat yield. There's a number of other potentials, and we're really excited about the opportunity for methionine to work post-absorptively to increase milk fat. There's great mechanisms at work that have been really well investigated in liver fatty acid transport and metabolism during the transition period. And we think some of those same mechanisms could be important to supporting milk fat synthesis during normal lactation. So like to thank you for your attention. The project was supported by Adisseo and Penn State. Also need to recognize the support we get from USDA and other sponsors in the lab. So happy to take any questions.
Thank you, Kevin, for your excellent presentation. I mean, this is a really full circle that we went from the beginning to the end now, and we're back inside the cell of the cow for what you're trying to say now in terms of the impact of try to elucidate what is affecting this milk fat depression. We always, we're thinking this is just this lumen effect, but obviously something else is going on. Now, you did mention that there were some, you did study some microbial effects. Can you expand a little bit on that, or do you have anything to make more comments about it?
Yeah, so the microbial effects were not in this experiment. They were in another experiment with HMTBA. So we originally had gone in to characterize what we call our favorite 12 microbes. So there'd be your common culturable microbes like Strep bovis and Fibrobacter that everybody recognizes, and we can see those change during milk fat depression when we induce milk fat depression, but we were not able to see an effect of HMTBA on those. What we did find with real-time PCR is an increase in protozoal abundance, and then we went and worked with Dipti to do the microbiome analysis. In talking about complex data, you're looking at everything that's there, right? And then you see a lot of different microbial species. There's a couple microbial species that microbiologists look and see their favorite guys changing and kind of get excited about the possibility of this one or that one. I will say that what we see is really a subtle change. It's not a sledgehammer effect. But I think that's actually interesting that HMTBA seems to be maybe targeting what are some important populations. I think we're, with that you have a characterization, so you're saying these guys are changing at the same time that you're seeing your production change, but it's really hard to say which ones of those are the important ones that are changing. But the biggest kind of global thing is that increased diversity, that there's a couple different metrics of diversity, and you seem to be maintaining more diversity. Now, why that is, I don't think we really know, but there is a positive effect there.
And so going on the other side now, there is another question. Is okay. So how does rumen protected methionine now prevents milk fat depression? What would be how would you explain that?
Yeah, so that's where I'm really excited about the possibility for post-absorptive effects. So I think we could think about there's well-documented effects on liver metabolism. So there would be the potential that you are assisting in fatty acid packaging and transport that would be making those fatty acids available to the mammary gland. I think there would be all sorts of what I think of as sparing effects. So if you're changing liver metabolism in a certain way, you change the nutrients that are available to the mammary gland because they have or have not been used by the liver. There could possibly be some direct effects within the mammary gland. I don't think those are as well documented as what we understand in the liver.
Yeah, and I think this is again goes back to the availability of these fatty acid precursors and what you're trying to, and what you were saying there, this sparing effect is really interesting area of work that we need to focus in. But I really thank you everybody for sticking in into this. I don't want to go much longer. I really thank you, Kevin, for your excellent presentation, and all of the other presenters. Just as a reminder, these presentations have been and are recorded. You would have access in feedchannel.online. And with that, I hope to see you guys next week. Thank you very much. Have a good day.